Good evening, everyone, and welcome to the April nanoTalks. My name is Joel Lutti, and I will moderate this evening's event. I'd like to welcome all regular nanoTalks visitors to this live stream and give a brief introduction for all the first-time nanotalk viewers. The nanotalks are a monthly event that Reach is organizing. And they're normally not held in my living room. They're normally held at the University of Zurich in an in-person event. In the nanotalks, two young researchers give a short presentation about their area of research and why it is important.
Good evening, everyone, and welcome to the April nanotalks. My name is Joel Lutti, and I will moderate the...
Sorry for this feedback here. Two young researchers give a short presentation about their area of research and its importance, followed by a Q&A and feedback from the audience to ensure that this talk is a good training opportunity for the young researchers. The nanotalks are organized by Reach. Reach is a young Swiss think tank that is really working towards a science-friendly culture within Switzerland, which means that we want to have science and society work side by side. If you want to learn more about us or join us, go to www.reach.ch. You can see the website here on the screen. We're mostly active in Zurich, Bern, Basel, and Lausanne with over 200 members. Come and join us for interesting events. Or sign up to our newsletter. You can also sign up by going to the link in the description of this video and go to the event page where we have a nice little box where you can sign up to stay informed on the nanotalks and on other Reach activities. Before we start with the actual content, I mentioned in the beginning that normally there's a Q&A and feedback, and we want to ensure that even in times of COVID, where physically you need to be distant from the speakers, you can still interact with them. For this reason, we're using a tool called Slido. Slido is an online tool for you to ask questions, and I'll give you a short demo on how to use it for tonight's talk. So you will find the link in the description of this video here, www.sly.do, and enter code nano-synbio. This will get you to the event where you can ask questions, and where later in the evenings I will open the polls. If you're on a mobile device, it will look more something like this. To ask a question, be sure that you're in the Q&A section, not in the polls section. And after the talks are finished, I will open the polls so you can give constructive criticism to the speakers and to us as organizers. To talk about how this evening will actually go, we'll first have both the talks of the evening, and then and Alexander is live here with me. Carolyn recorded her talk this morning, and then Carolyn will join us in a Zoom meeting so we can do a live Q&A with the audience. The first talk will be by Alexander. Alexander is studying biology at the University of Zurich, and I actually know him personally because last summer he spent the whole summer in the Pelkmans lab where I'm doing my PhD. As an undergrad researcher leading the iChem team of the University of Zurich. And he's also the president of SynBio UZH. So he's a great person to talk more about the promise of synthetic biology. Alexander, the floor is yours.
Yeah, thank you, Joel, for the great introduction. And hi, everyone, also from my side. In my talk, I want to talk about the biggest global issue that we are all facing today, which is climate change. And I want to take you all and see if we can, or how sustainable fuel can provide or can be used to address this global issue. So first I want to cover some common ground. The global average temperature has been rising since the past 100 years, and the main contributor to that is the increase of carbon dioxide in the air. Since the Industrial Revolution, around the early 1900s, we've put so much CO2 in the air that it's risen above any levels that we have seen or we have been able to observe in the past hundreds of thousands of years. And this doesn't stay without consequence. I think we're all aware that the risk of climate change is very high. Increased droughts, floods, and frequent strong weather events, these are things we're already seeing today. So imagine what it will look like in 50 years. And most of people, or many of the people who are listening right now, and certainly our children, will live to see this day. So I think we can all agree on these three things: that the global temperature is rising, it's caused by our human activity, and that we need to do something now. And based on which polls you kind of look at, you can find support for these three things globally in many different countries. So that's where we're all on the same page mostly. But where we disagree on is how actually do we fight, or do we do something about climate change. And the reason why we differ is because there's so many ways we put CO2 in the air. There's also many ways to prevent this from happening. But we kind of have to start somewhere. So I want to look at where does the CO2 globally come from if you divide it by very broad categories. So these are the CO2 emissions by sector. You see that energy production takes up most of it because a lot of the energy we produce comes from fossil fuels. Then the next two biggest chunks are transportation and industry. And I can't, in the next 12 minutes that remain, explain to you or show you a way how we can reduce the CO2 emissions from all of these sectors to zero. That would be a bit ambitious. But I can show you how it's possible to do it for this chunk here. So transportation puts CO2 in the air by burning fuel. So cars, planes, and ships, they all use gasoline. And I think it's obvious to everyone who's watching that this fuel is not sustainable. The fossil fuels in the ground that we turn into CO2 in our engines, this CO2 never goes back to fossil fuels. It's not a sustainable cycle. And now I want to look at the alternatives that we have for a more sustainable kind of fuel, so that we, but with the strict condition that the alternative has to have a zero, or basically no additional, that the alternative puts no additional carbon dioxide in the air. That's the condition. So now, which would be the most obvious alternative if you think about it? Most people will probably think of electricity. It's a type of fuel. You can read up on that. And electric cars are great because they don't use any fossil fuels, so they don't produce any CO2. The only thing they need is electricity. But where does the electricity actually come from? 65% comes from fossil fuels. So although your car isn't putting out any CO2 directly, the energy that is required is still produced from largely fossil fuels. So this is really not an option for what I'm trying to do, or we're trying to do today. So we'll have to look for something else. So what other options are there? My dad would now say, listen, hydrogen cars are the future. So, all right. Hydrogen-powered cars don't need electricity to charge their battery. Instead, the cars contain a hydrogen tank where you can fill it up like normal fuel, and then the hydrogen goes through a chemical reaction and energy is created. So no fuel and also no electricity directly, but the hydrogen is actually produced from natural gas in a process that requires large amounts of energy. And the energy comes from mostly fossil fuels. So again, this is not an alternative where we have zero CO2 that's in the end produced. Now, the third option is maybe not so well known, and it's the ability to create biofuel, which is basically like normal gasoline from plant biomass. So this can be fuel, oils or fats, or just rotting plant material, which is a nice idea because you have like a cycle. The biofuel gets burned, but plants can take up CO2. However, the issue is that this conversion process requires a lot of energy, and the energy largely comes. So I think, so do you see what I'm trying to do? I'm making a point here, and my point is that we think that our fuel is not sustainable, but actually the issue is that the way we produce our fuel is not sustainable, right? Because to produce fuel, you need energy. But the whole energy system on our planet is based on fossil fuels. So we can't really detach the fuel production from the energy production, and the energy production is not sustainable. And that is really the issue we have to overcome for alternative fuels, right? To detach fuel production from energy production, which doesn't really seem possible. So the only option really seems to be to completely change the way we produce energy and just use renewables or nuclear energy to really drop our carbon emissions to zero from fuel. But before we consider or we go to this way, I want to show you one more alternative, which is a totally different process from any other way to produce fuel. And it's schematic, but I'll guide you through. So the idea is to create a factory, and the factory is supposed to be able to take CO2 from the atmosphere, and then a process called carbon fixation creates biomass. The process carbon fixation needs energy, but by a state-of-the-art converter, we can turn sunlight into energy, and this is not a solar panel that I'm talking about. The biomass can then be in a second factory converted into something like biofuel. And this conversion process doesn't need any electricity. Biofuel can then be used in your regular car, will be burnt into CO2. However, this will be again taken up by the first factory. So in the end, you're not putting any more CO2 in the atmosphere. And this process would be able to create fuel completely detached from the energy production. That's not sustainable. And I think, people who know me or who are biologists, they kind of see what I'm doing here. So the first factory is just a plant, or more specifically, it's a plant cell. Every plant is able to take up CO2 from the air, and through specialized organelles, the green ones here, called chloroplasts, they can turn sunlight into energy. With the CO2 and the energy, the plant can build up all that it needs, sugar, leaves, roots, and all that, and that's what we call biomass. Now, the second factory is actually a bacteria, also a living organism. And some bacteria can, in a process called fermentation, turn organic mass, like plant material, into things that are very similar to biofuel. So this is really what we're looking for. We don't need any electricity from the outside, and we have a sustainable process to create fuel. Now, this was a bit of a weird way of introducing you to plants and bacteria because I was showing these factories. And that is the viewpoint of a synthetic biologist. And it's not something that's unfamiliar to you. Think back when you learned about biology or when someone told you about what a cell is. You probably looked at a picture that looked like this. So this is how we teach about cells. you have to kind of imagine it like a little factory. So a cell can take up some nutrients, and in some kind of, let's call it assembly line, the gray and the green line, the plant can take up nutrients and turn them into something that the plant or the cell needs to survive. For example, sugar or any other material. And the synthetic biologist now goes a step further and realizes that we can not only view the cell like this, but we can also understand it in this way, that it's a factory, meaning it's composed out of interchangeable parts. If we just understand it well enough, we can, for example, change the assembly line so that the cell can create something it's not normally able to. For example, biofuel. So I want to get now a concrete example of how one could actually take a cell that's not able to produce biofuel and turn it into a cell that can. And the specific example will be the production of biodiesel with microalgae. This is one of the more, let's say, realistic or better ways to produce a type of biofuel, because there's a huge variety. And for this process, you need two things. You first need some kind of oil and ethanol. This particular microalgae here has a very high oil content, or can produce very high amounts of it. And alcohol or ethanol, well, if you drink beer, you should know that beer is brewed with yeast, so yeast are very good at producing ethanol. Now we just have to take a bacteria, and this is supposed to be able to turn these two ingredients into our biodiesel. Now, as I said, producing ethanol from yeast is easy. The microalgae part is a bit more tricky because there are some restrictions on the growth, etc., but it's doable. The really difficult part is this here, because no bacteria that exists today can actually produce biodiesel from just oil and ethanol. And why not? Because it's missing the assembly line to do that. So this is something we have to create now. And in the very first step, and I just want to show you the very first step in how you would actually genetically engineer this. So these will be our starting materials, ethanol and oils, which let's call them fatty acids. So we have to turn these into, in the first step, a biodiesel precursor. And this can be done by something called an acyl transferase. Now you don't need to know what this is. Just think of it as an assembly line that can turn these two products or these two compounds into our precursor. Now we can understand, or we can do this process chemically or biochemically in a test tube very simply, but we want a bacteria to do it for us because that requires way less, or that's the way we produce it, because we don't want to do the work. The bacteria should do the work. So what we have to do now is this assembly line needs to kind of go into a bacteria. And for this we will use E. coli. It's the best studied bacteria that we know, and it's been genetically engineered and modified already. So if you want this E. coli to have this assembly line, you need to provide it with a blueprint of it. That means a chunk of DNA that the bacteria can understand, so that it can create the assembly line in its cell and produce, or will just go through this reaction. Now you have to do quite a lot of thinking on that, because many different of these acyl transferases exist in nature, and not every blueprint is readable by the E. coli, and so this really requires a lot of forethought. And in this way, you would think about each and every following step until you get to your biodiesel. And that's how you would engineer an E. coli to produce biofuel. Now, let's take a step back from this very detailed look at how to do this. I kind of want to mention a very important aspects of this particular fuel alternative, which is first that if we use biodiesel as in contrast to electricity or hydrogen powered cars, we can keep the existing infrastructure, because most cars could actually run on this biodiesel. And developing countries don't really have the money to invest in electric cars and all the necessary battery production and energy storage. And also, it's a very good for branding, because you can brand it as a kind of green fuel, and very new, very hip. So that's also something you have to consider. That's valuable. Now, the big question is, can we actually do this? Is the biology ready? And the answer is yes, because this exact process, what I walked you through, has already been done, actually by a couple of labs. But the issue is, it's been done in a test tube in a lab with a few milliliters. But to actually make this work, you would need to scale it up to this level. And that's the main key, or the main issue for every synthetic biology application. How do you take it from the lab to a scale where it's actually profitable? And that's has a lot of limitations still. So to finish, like, will this process be revolutionizing transportation in 5 to 10 years? Probably not. And that's fine, because that was not the point that I wanted to make today. It's not just about the biofuel. I wanted to remind you of this viewpoint of viewing cells like bacteria, like algae, as factories. And I want you to take a step further and think of them as changeable, so we can modify these cells to do whatever we want them to do. This can mean producing antibiotics, or anti-cancer drugs, biofuel, or even things like spider silk. And all that without the need for electricity that's coming from an unsustainable process and from fossil fuels. And that's really what I mean with the promise of synthetic biology. And that's it. Thank you very much.
Thanks a lot, Alexander, for this really interesting talk and this pitch for synthetic biology. Can I take the seat?
You can take the seat.
So thanks for staying with us for this first talk. We have a second talk upcoming. Before we start that, I'd like to remind you again that you can participate in the Q&A by going to sly.do and following the link in the description of the video, where you can ask questions like the 4 that have already been asked. And then after Caroline's talk, we will actually answer those questions in the group. Now, for the second talk, for Caroline's talk, we're making full use of this remote and stream talks now, because normally we couldn't have a speaker from Oxford at a nanotalk at the University of Zurich, but now that everything is happening online, it's my great pleasure to have Caroline Schulte for the second talk. Caroline is a PhD candidate at the Department of Engineering at the University of Oxford, and she recorded her talk this morning, so we could show it now here, and she will join us after the talk in a live Zoom Q&A. I've one interesting fact about Caroline. She actually founded a startup called Inulux at in her first year of her PhD. And the startup works on enzyme-based supplements that reduce absorption of dietary sugars. So I think we really have someone here again who can tell us more about potential of synthetic biology. And now I will play the talk of Caroline about biological nitrogen fixation for you.
So good evening everyone, and thanks for joining for my talk. My name is Caroline and I'm a PhD student at the University of Oxford, where my research revolves around plant-microbe interactions, and as you can see from the title, it's about a process called biological nitrogen fixation in particular. So for my talk, I would like to explain the science behind biological nitrogen fixation and also outline the limitations of this process. I will then briefly describe the alternative that humans have kind of come up with to address those limitations. And finally, adding on to Alexander's talk, go into the solutions that synthetic biology is trying to develop currently in order to overcome the limitations of biological nitrogen fixation in a sustainable way. So we're starting with the concept of biological nitrogen fixation itself. If you look at plants grown for agricultural purposes, it is very important to determine the limiting factor of plant growth. Because essentially, while we have carbon dioxide in the air, sunlight and water, which are usually supplied in sufficient amounts, phosphorus and nitrogen are commonly the limiting nutrients for plant growth. And while I will not go into phosphorus, the interesting thing about nitrogen is that it's actually abundant in the atmosphere. So N2 gas is about like 78% of the air we breathe. However, in this form, it cannot be used by plants. So it would need to be assimilated into a different chemical form, ammonia, for the plants to be able to use it. And nitrogen in itself is just a very important component of most biomolecules. So for example, the genetic material, DNA, present in every cell, requires nitrogen, but also proteins, which ultimately form a very important part of the human diet, require nitrogen. And to basically overcome this issue of not being able to use nitrogen that is present in the atmosphere, a certain plant species or family of plants called legumes, which comprises peas and beans, for example, has evolved a symbiotic relationship with certain soil bacteria called rhizobia. So the way this works is schematically displayed here on the right. Essentially, the soil bacteria, the rhizobia, induce the formation of nodule structures on the plant roots. And inside those nodules, the rhizobia will receive carbon from the plant, and in exchange for that, convert the atmospheric nitrogen into ammonia, which the plant can then use. So I have just briefly illustrated this here, where on the left you can see two plants, the left one not having rhizobia on its plant roots and obviously being nitrogen starved, while the one on the right has formed this symbiotic relationship and continues to grow. And on the right, essentially, you can see a close-up photograph of nodule structures on a pea root. So essentially, you've got those pinkish structures, which are the nodules, and inside those you would find the rhizobia, which are fixing nitrogen. So this symbiosis, while being very beneficial, is a very complex process, which is still not fully understood. So just to give you an idea about like just the formation of symbiosis, you have to see that the soil microbiome is essentially very complex. You have an abundance of different microbes which can be beneficial, such as rhizobia, but also pathogens. And essentially the plant needs a way to decide which bacteria to let inside its root. So the way this happens is usually through a complex exchange of signaling molecules which the plant secretes under nitrogen stress, and which are then specifically recognized by rhizobia, which can then respond in a form that the plant recognizes. So shown here on the right is once more like a scheme of the symbiosis formation, where the bacteria essentially need to be attracted to the plant root and then induce this root hair curling. They're kind of entrapped by the root and then grow down into the plant tissue, where they differentiate into the nitrogen fixing form called a bacteroid. And these bacteroids are very different from the free-living bacteria, so they completely change their metabolism, stop dividing, and essentially just become little nitrogen fixing factories for the plant, essentially. So while this process is obviously amazing and hugely important for agricultural yields, it unfortunately also has certain limitations, as shown on this slide. So as I mentioned earlier, just over the course of evolution, this process has only evolved for legumes, while major crops such as wheat, for example, or corn, are unable to form a symbiotic relationship with rhizobia, and thus, hugely suffer from nitrogen limitation. The other thing, which is more related to properties of rhizobial strains, is this issue of competitiveness and efficiency. So what I've schematically shown here on the right is that you might have strains such as the one shown in this brownish color, which are really good at getting to the plant root and forming nodule structures. So they're highly competitive, but they're not very good actually at fixing nitrogen, so they have low efficiency. So this conflict of symbiotic partners, as I've written here, essentially refers to the fact that it's not in the interest of the bacterium to fix as much nitrogen as possible. But for the plant, it's obviously hugely important because it has to invest carbon into nodule structure formation and maintenance of its symbiont. And so yeah, for the plant, it can actually be kind of detrimental if an inefficient strain forms nodules and the plant has to maintain it. So as you can see from the slide, yeah, biological nitrogen fixation has huge limitations if we want to intensify agriculture, as we're currently doing. So humans have kind of come up with their own process, which I've just termed in quotation marks, human nitrogen fixation, which is essentially a chemical process to achieve the same thing. So just by means of a brief motivation, I mean, we all know that the population continues to grow, essentially predicted to reach 11 billion by the beginning of the 22nd century. And at the same time, we're faced with decreasing area of land that we can actually farm due to the effects of global warming, which causes here spread of deserts, but also flooding and erosion. So what humans come up with is essentially this chemical process called Haber-Bosch process, which converts atmospheric nitrogen into ammonia, and has been one of the key enabling technologies of the so-called Green Revolution, which essentially enabled great intensification of agriculture and had enormous consequences also on a social and economic level. So as you can see here, while the area of land that we're using for agriculture has not increased much since the 1960s, the yield that we get, so the crops per area that we're using, has risen dramatically. And this has been hugely empowering, especially for farmers in developing countries, which through access to fertilizers were able to first of all supply food for themselves, but also be able to sell it to make a living from it. As usual, it's not all positive. It would be too good to be true, because the massive issue we're facing with fertilizers are kind of twofold. On the one hand, most of the fertilizer that is applied is actually washed out into the environment. So you get this eutrophication of aquatic systems, where essentially nitrogen leakage into the environment causes algal blooms, and then consequent death of fish and other organisms. On the other hand, the Haber-Bosch process in itself is very energy intensive, so it requires those extreme conditions, and essentially accounts for around 2% even of the global energy demand every year. On a higher level, kind of by massively applying fertilizers to crops that we're planting nowadays, become heavily reliant on the fertilizer application, so they essentially lose their ability to form symbiotic relationships with bacteria. And then I mentioned the economic benefits of fertilizer application. However, due to this high energy demand and the requirements of the production process, fertilizer is still quite costly. And some people actually argue that it has rather increased the gap between rich farmers in developed countries versus the very poor who cannot afford it in developing countries. So obviously, we're not quite there yet. We need another solution for high yield but sustainable agriculture. And this is kind of where synthetic biology comes in to improve on the natural process of biological nitrogen fixation. So what I've shown here is, first of all, on a very basic level, I mentioned efficient and competitive strains. So first of all, to find efficient ones, what I'm doing in my PhD research is essentially combining computational and experimental tools to understand the metabolism really of bacteria when they're fixing nitrogen, to then identify limiting steps essentially and see how we can engineer strains that are just more efficient at fixing nitrogen. On the other hand, as I mentioned, we also need strains that are actually really good at forming nodule structures on the plant root. So recently in my lab, actually, a tool was developed where different strains can be marked with this green fluorescent marker. And then basically you can put all strains on a plant at the same time. And obviously the competitive ones will form nodule structures. But then the readout that you get, the intensity of the green fluorescent signal, is proportional to the efficiency of the strain, so how much nitrogen it fixes. So you kind of have a nice high throughput tool to evaluate which strains are competitive and efficient at the same time. On the other hand, looking at like true synthetic biology solutions, there are several approaches which are being investigated in parallel because we do not know yet which one of those will ultimately work, hopefully. So first of all, an idea would be to transfer this process of nitrogen fixation into the plants directly. So to overcome the limitation to legumes and also just not be reliant on the bacteria anymore. However, this is really complicated because nitrogen fixation requires a lot of proteins, cofactors, which are very difficult to produce directly in a plant and requires the transfer of many genes into the plant itself. Another approach relying more on the natural system would be to engineer synthetic symbioses. So to take a plant such as corn, which does not form the symbiotic relationship, make it produce certain signals that are then recognized by rhizobia, which then respond and so form this artificial symbiosis kind of. Along a similar line are kind of the free-living fixers, so nitrogen-fixing bacteria that do not normally form symbiotic relationship with plants. So normally they would only fix nitrogen for their own benefit. And so those would not require the formation of nodule structures. However, we need to find a way to essentially make them secrete the nitrogen that they fix and make it available to the plant. So yeah, those are currently the main strategies that are being pursued. As I said, it's not clear at all yet which one of those will eventually work. But I hope that I've given you a good overview of where we're at in terms of working towards a more sustainable agriculture in the future. With that, I would essentially like to thank both of my groups that I'm part of here in Oxford, and I would be happy to answer your questions now.
And with that, we're back and setting up for the Q&A. While we set up for the Q&A meeting with Carolyn, you still have the chance to get in your questions. Do this at slide.do, code nano-synbio. And post your question here so we can then answer them afterwards. So let's give Carolyn a minute to join. Okay, and Carolyn is here. Hello, Carolyn.
Hey, how's it going?
Yeah, all right, thanks. Well done for your talk.
Thank you. Yeah, same to you. It was quite enjoyable.
Yeah, we had 2 really interesting talks about SynBio here. Thanks a lot to both of you. We have quite some questions from the audience, and I will read some, and then sometimes they're clearly to one or the other person. Maybe there are some you might both want to say something about. Just chip in and discuss. So maybe the first question, most upvoted question at the moment is for the first talk for Alexander. And the question from an anonymous viewer is, why can't we use ethanol and oils directly or find a chemical process to turn them into usable fuels? Why use the bio factories?
So that's a great question because it really strikes at the core of the whole issue. You can do it in a chemical process. The issue is the amount of energy required is quite high. And now any engineer or person who is knowledgeable about thermodynamics will say, yeah, but you still need the bacteria also still need energy to do this process. But in the bacteria, you have things called enzymes, and those can basically greatly lower the energy that is required to do the process. So you basically, a lot of the energy that's required is outsourced to kind of the, let's say, complexity of the bacteria, if you want to call it like that. So you can do it, but it requires a lot of energy. And then the bacteria, this is kind of saved in a sense, if you get what I mean.
Okay, so basically we use the bacteria to do it more energy efficient than if we would do it in an existing factory kind of setup.
Yeah, that's exactly it. It's more efficient. Yeah.
Okay. Thanks, Alexander. Maybe a question for you, Carolyn. There was one question you said in the beginning. Let's not talk about the phosphorus. Let's focus on the nitrogen. Antonio, it's an anonymous person or viewer wanted to know. Well, if we do this, how do we then solve the phosphorus problem? Because that probably then becomes a limiting factor, no?
Of course, yeah, great question. I mean, it obviously depends on the area where you're working, the kind of soil that you encounter, which limitation is more prevalent. I mean, often you have both, but sometimes you just have one or the other. And essentially, there's also phosphate solubilizing bacteria, so bacteria that secrete certain enzymes or organic acids that make the phosphate, which is usually present in the soil, but also not available to the plants because it forms certain complexes and can't be assimilated. But there's also bacteria essentially able to make the phosphorus available to the plant. And this is a whole different area of research, which I'm personally not working in, but which is also, of course, highly important for agriculture.
And where we could also use a SynBio approach eventually.
Oh, absolutely. I mean, that would be kind of the dream to have like the super bug if we can and at the same time secretes like those phosphatases that make the phosphorus soluble. Or even better to have like a bacterial community where you have like one really good at fixing nitrogen, the other one really good at solubilizing phosphate, and the two kind of synergistically interacting.
Okay, cool. So certainly something that could be tackled with those same approaches then. Maybe talking about having communities of microbes, I think that question is more back to you, Alexander. And the question from another anonymous viewer was, how would you tackle opportunistic mutators in a bioreactor that lack the assembly line function, but just consume your inputs and thus have an evolutionary advantage and just grow fast?
Yeah, so you really get into a lot of details now. That's a great question because I haven't considered it at all, but I really see where you're going with this. So the problem would be that, let's say, just to rephrase it, you have a lot of microbes or a lot of algae or bacteria or whatever, and if one of them mutates and has an advantage over all the others, it doesn't contain the assembly line, it would just outgrow everything and you would just be left with that. What you could do is you could tie basically having the assembly line, you could tie to some kind of survival factor in the bacteria. That's basically rather standard practice in synthetic biology. You have things linked to either death or survival factors. So if you lose something or gain something, this can mean the death of the organism. Of course, you can play this game where the bacteria can also overcome that. This is really a question for the future scientists who have to tackle this issue, but I hope you kind of see what I try to answer, or what I propose with my answer.
So it's not, what you're telling me is it's not like research in this area isn't finished yet. So there's still work to do for PhD students and potential future students in this area.
Yeah, I mean, for sure, yeah.
Yeah, okay. But good for bringing this up then. Seems to be an interesting issue here. Let's go back to you, Carolyn. And there was a question about soil bacteria. And an anonymous viewer wanted to know whether you think that we could make soil bacteria, or there even are soil bacteria at the moment, that would have a potential to draw just CO2 from the air to fix it, and whether this could be done at any kind of scale that would have an impact on climate change.
Yeah, it's a great question. There are definitely rhizobia, so nitrogen fixing bacteria that can also fix carbon dioxide. The particular species that I'm working with cannot do that, so I'm not an expert in that area. Also, mainly actually, if you think about carbon dioxide fixing bacteria, that would be cyanobacteria, so kind of like a different family. I must admit, I do not know in terms of quantitative impact how much that could actually do in terms of reversing climate change. I have not read any studies about it, so I would guess at least that the main benefit at the moment of rhizobia is the nitrogen fixation rather than carbon dioxide fixation.
Can I just throw in there just quickly that the soil is actually the biggest sink for carbon dioxide. So it's not forests, it's not oceans. The soil is what holds the most CO2 bound in that sense. I'm not sure if it's this because of the biomass or because of minerals, but maybe you could also look into that, and from that maybe find your answer.
Okay. Thanks for this shared answer. That probably also just answers the question that Dominic had on a similar topic. Another thing you brought up I think especially in your talk, but I think it also came a bit up in yours, Carolyn, is the question of like scaling this up and doing it at a bit larger scale than small experiment. And an anonymous viewer wanted to know, why is this scaling up so hard? Like, why did you say, scaling up of this is really hard?
Okay. So if you think about, you have a bunch of algae that grow in a small test tube. The light can reach all the algae pretty well. But if you want to scale it up into a size of a couple of cubic meters or even more, if you just have a huge tank, the algae in the middle won't get enough sunlight to actually be efficient. So you have to devise a way where you have like kind of the maximum amount of cells in a sort of good like wide, so that all of the cells get like the best amount of sunlight to be maximally efficient. And just this kind of process of figuring out, okay, what works best requires a lot of probably mathematical modeling and a lot of trying things out, and that is a huge issue. And also things like diffusion, how are you going to collect all the product that is created efficiently, so that it also doesn't impact all of the other cells, working away. So it's quite tricky.
Okay, so really like creating an environment.
I
Carolyn?
throw something in there as well, because I actually did my undergrad in process engineering, so I did quite a lot of maths behind that. And the thing is just, as soon as you go like into a big bioreactor, it's not just a shaped flask times factor X, because the forces you have in there in terms of oxygen diffusion, sharing forces from the mixing and everything are completely different. Also, like, the key thing that all process engineers like to talk about is the surface to volume ratio. Essentially, you run into massive cooling issues because microbial metabolism produces a lot of heat. And as I mentioned earlier, especially getting enough oxygen in there for aerobic cultures is usually the main issue.
Okay, so it's really interesting engineering challenge then on how to scale something up that works once in the lab.
Yes.
Maybe staying a bit on in this kind of topic, and like how do we actually then apply it? There was a question from Antonio. The question was, what are the required growth areas for growing, for example, algae? And he wants to know, is this a way to avoid conflicts with agriculture for nutritional purposes? Or would we have competition on where we grow synthetic biofuel compared to our agriculture?
Right. So actually, I read about this in a couple of reports where this was estimated, and what I saw is that for the US, these are just some estimates, if you want to replace the transportation system completely with biofuel generated from algae, you would need about 0.24 to 0.1 to 0.5% of the total surface of the US which is about 1 to 3 times the size of Belgium, which sounds like insane, but you have to consider that that's only 1/7 of the size of the area where corn is grown in the US, and most corn is actually fed to livestock. And you can build you can grow algae on non-arable land that you wouldn't use anyway otherwise. And the big advantage is that you can grow algae not just in fresh water but also in more salty water, wastewater, and actually create, potentially some startups claim that, fresh water. So I hope that answers your question.
Yeah. Okay. So that basically means there shouldn't be much of a competition, because you could really switch to places where we are not currently growing corn, for example.
Yes, but it would introduce the problem of how do you after that move all of your products? Because the amount, I mean, if you want to supply the whole US with energy, it's a logistical challenge. That's also an issue.
Maybe like on the same spirit for you, Carolyn, would those synthetic biology approaches of creating nitrogen fixation, for example, with synthetic biology, would that allow us to use new areas for agriculture as well, or?
Oh yeah, absolutely. I mean, that's what I mentioned earlier in terms of how the green revolution, like the application of fertilizers, just allowed people, for example, not even just Africa, but also Australia, for example, have massive issues with very poor nitrogen poor soils. And so there's definitely a very enabling technology. And the nitrogen fixation, in contrast to fertilizers, just allows us to do this like in a sustainable way and potentially more affordable. A great advantage also, like if you have those rhizobia, is actually, again, in contrast to fertilizers, they tend to stay in the soil. So actually quite a few decades ago, before people even figured out the science behind it, they did this thing called crop rotation, where you would first plant like peas, beans, and lentils, and after that the soil would be nitrogen enriched. So if you then plant wheat, corn, whatever, that just grows better. So yeah, definitely that will allow us to use new areas, but also just use them more intensively, get better yields from them.
And still keeping our waters cleaner than if we use more traditional fertilizers, I assume.
Yeah.
There's some other question more towards the efficiency, or I will combine two of the questions here. One of them, I think both for you a bit, Alexander, is a bit about energy efficiency, how energy efficient is like a biofuel production compared to using batteries. And like what kind of inputs do you need? Do you also need some kind of sugar as an input for your yeast, or like how much energy do you put in? How much are you getting out?
Yeah, that's a very important question. So for the yeast, you're actually right, and this is something that I realized kind of when I was already done with my talk, basically set up and everything. Yeah, you know, the yeast also, of course, needs something to produce the ethanol. So that's certainly an issue. But you could again say, okay, we use plants that make biomass, and from the biomass we make ethanol through bacteria or through yeast. That should be fairly easy, I think. To the efficiency, so that's something that I really didn't look into much, which is a shame because I see the importance of it. So that's not something I can really answer confidently. Sorry.
Well, thanks for the answer that we got anyway. Maybe another question for you, Carolyn, from a viewer is, you describe basically, we can create this bacteria that does the fixation. Could we use the same bacteria everywhere, or is the background soil community, would that affect how efficient that process would go, or how that symbiosis then works?
That's actually really amazing, like really close to current research efforts, because while like most basic research was done in like very sterile systems where you just have your single strain, we're actually increasingly recognizing how the existing soil community that you put your strain into massively affects its performance. And first of all, as I mentioned in my talk, you get like strains that just outcompete the others. You also get pathogens. And yes, soil communities are just like an incredibly complex ecosystem. There was a recent study where just like 7 strains, which is like very simplified even, were put together, and then they one by one just removed a single strain from the community. And there was like a certain like enterobacterium, for example, when you removed that, just the whole community collapsed. It couldn't reach like a stable equilibrium. So you can just see how the soil, like not just the nutrient composition, but also the existing microbiome, has a massive influence on how your synthetic inoculum will perform.
Okay. Again, a bit of the point of it is a complex field, and there's certainly more to study in this field.
No, we definitely haven't figured it out yet. I mean, other approaches are rather like enhancing what is there rather than putting something else into a community that we don't understand in the first place. But yeah, the answer is we are still trying to figure it out.
A bit on this, so to finish this Q&A now, a bit on this topic of there's still things we want to figure out, and maybe to give you a bit of a chance to pitch what you're doing. If people want to learn more about this, if people want to get in contact with you or follow you somewhere, what would they do? Where would they go if they want to hear more about the synthetic biology in Zurich or in Oxford? Maybe, Alexander, you want to start?
Yeah, I mean, I'm not sure if you've noticed, but I'm wearing this nice sweater here. If you're interested in synthetic biology, the best place to start is probably the website of our student organization, which is SynBio everythingtogether.com. So just pulling it up now. And I also suggest you to look at the iGEM team of the University of Zurich, which we started last year. Now there's the second team, running in this. It's the largest competition in synthetic biology, and you can follow their journey on Instagram at iGEM-UZH. That's actually from our student organization.
Okay.
But yeah, I mean, this is the place where you can really go, and you can also send me an email. My contacts info is also is here. So get in touch, and we can talk more about these things if you want.
Thank you, Alexander. And Carolyn, do you want to pitch something?
Yeah, I mean, if you're interested in like plant microbe interactions, have any questions there, I had the two groups I'm affiliated with on my final slide, I guess, so they're reasonably easy to find.
I can bring that up again.
Other than that, I'm a member / co-founder of the Synthetic Biology Society in Oxford, which is synbio-oxford.org on the web if you're interested. I'm actually hosting quite a few webinars at the moment due to the current situation, so everyone very welcome to join. Other than that, I'm actually a member of the iGEM entrepreneurship program as well, which is how I initially reached out to Alexander. So if you're interested there, it's called iGEM Ethic, like Entrepreneurship Program International Committee. So yeah, we're always happy to receive application on any questions. Yeah.
That is very cool. And then I want to really thank you. Thank you for taking the time, giving this nice presentation here. And I would also like to pitch where you can find more about Reach and follow future nanoTalks, which is basically follow the link in the description of the video. You will get to the event page, and on that event page we have a newsletter sign up form. Sign up there and you'll hear when we have more nanoTalks. The way the situation is currently developing, we're also planning to have more live stream nanoTalks. The next one will be the last Thursday of May, which I think is May 30 or so. Actually, we'll have to quickly check. Don't have that live at the moment. And May 28. And that will mostly be on the topic of water and water sustainability. So join us then again on live stream nanoTalks. Thanks both to you, Alexander, and to Carolyn for joining tonight, and to all the viewers. Have a good evening and stay safe.
Bye. It was a pleasure.
Questions as well.
Bye.
Bye.